Thanks, Michelle. First of all, thank you to Michelle and Joanne for organizing this event, and thanks to those of you who made it out this early in the morning. I suspect there's some CRISPR meeting fatigue, perhaps, especially in this area of late. So I'm very pleased to be able to tell you about some of our recent work, and also I'm hoping in the last third or so of the talk to tell you about some even more recent work that is not yet published as well. So I don't seem to be able to advance the slides. So if I could get the next slide, please. So I'm required first to show you this slide by my institution, which is simply to point out that I may have potential conflicts because of my association with the companies that are listed here. I am a scientific co-founder and consultant for Editas Medicine, which is a company that's trying to translate some of the technologies we're going to talk about today into human therapeutics. And I also am on the SABs of Transposition and Horizon Discovery, again, companies that are trying to commercialize for the research market some of the technologies we'll talk about today as well. So I've done my duty to my institution by showing you this slide. I always point out the great irony, of course, is essentially free advertisement for these companies. Who might think that was set up outside. So this audience probably doesn't need this slide. What is targeted genome editing? Well, it's just a technology that allows you to go into living cells and organisms and in a targeted fashion and with very high efficiency make desired changes of interest to the DNA in those cells. I seem to be able to do this. There we go. So how do you do this targeted genome editing process? Well, the initial event is a targeted double-strand break introduced into the sequence of interest. Then you rely on one of two repair pathways that are operative in most cell types and organisms. The first is known as non-homologous end joining, or NHEJ. This is a process where the two ends are simply rejoined back together. For the types of breaks introduced by the nucleases we're going to discuss, this process is error-prone, and so with pretty high frequency, you get the formation of insertion or deletion mutations that initiate half the side of the break. And so if you put the original break into the coding sequence, particularly early in the coding sequence of the gene, you'll get these variable length frame shifts introduced into that gene coding sequence, and therefore generate the high frequency knockout mutations. And this is the process that most people utilize in order to introduce mutations. There is a second DNA repair pathway that's operative in a lot of cell types and organisms, as well known as homology-directed repair or homologous recombination. Here, you're using a donor template, what's called a donor template. This is a piece of DNA. It's either single-stranded or double-stranded, and that is essentially identical in sequence to the region in the genome where you place the break. But it has some other desired change in its sequence, and that's shown here with the green dashes. So this can be point mutation or a series of point mutations or even insertions up to several kb in length. And so in this process, the break is repaired using this donor template as genetic information, and essentially this sequence is incorporated into the endogenous allele. So by this process, you can make precise mutations or alterations, and it also offers the possibility of being able to correct mutations back to a wild-type sequence, which of course is of interest for using these reagents for therapeutic purposes. But the challenge for many years, and really when we first entered this field now about 9 years ago, was how do you make this targeted double-strand break in the first place? And of course the answer is that there are, next slide please. The answer is that there are a variety of different platforms now available for being able to introduce targeted double-strand breaks into your sequence of interest. So in the early days of the field, people used meganucleases and engineered zinc finger nucleases. The challenge with these platforms is that they are more difficult to re-engineer their binding specificities or cleavage specificities. More recently, the transcription activator like effector nucleases or TALENs were introduced, and these offer the possibility of simpler, more modular assembly to be able to target a wide range of different sequences. And then even more recently, the introduction of the CRISPR-Cas9 nucleases that are guided to their sequences primarily by a DNA-RNA complementarity. And so, of course, this latter platform is the one that we'll focus on today. Next, there we go. And next slide, please. So how do these nucleases work? Well, so if you have a target sequence that you want to introduce a break into, you identify the sequence. It's typically about 20 nucleotides in length. And I'll show you in a second why the strands are separated. There is a requirement adjacent to this 20 nucleotides for an additional sequence known as a protospacer adjacent protein. So for the Cas9 nuclease that most people use, which is derived from strep pyogenes, this so-called PAM sequence needs to be of the form NGG and needs to be directly adjacent to the sequence here that you're targeting. So in order to target this site, you need to express two components. One is a short RNA of about 100 nucleotides in length. The 3' leading nucleotides of this RNA are constant and fold into a characteristic secondary structure. And what you do is you design the first 20 nucleotides or so of this guide RNA to be complementary to the complementary strand of the sequence that you're trying to target. And so this is just designed using standard Watson-Crick base pairing rules. The second component you need to express is that Cas9 nuclease protein. Cas9 complexes with the guide RNA recognizing the secondary structure here. It then is believed that this complex scans the DNA looking for PAM sequences, and then if this 5' end of the guide RNA matches relatively well to the target sequence adjacent to that PAM, then you get a cleavage event, which is a double-strand break, blunt double-strand break that occurs about 3 bases in from the edge of this PAM here. So in this system now, recognition is primarily driven by this guide RNA DNA hybridization interaction. Although again, a little bit of the interaction, or not a little bit, but part of the interaction is provided by recognition of this PAM sequence as well. So this now greatly simplifies the targeting of nucleases because you're just relying on that simple Watson-Crick base pairing complementarity. And this paper published out of the labs of Jennifer Dowd and Emmanuel Charpentier in 2012 really kicked off this whole field because it described this basic system here that I've just gone through with you and demonstrated that it worked in vitro. So in that paper, they were able to cleave 5 different target sequences in isolation in a test tube. And at the time it was published, the question I think that was on everybody's minds was, would it work in vivo and would it work in cells? And of course, the answer to that is that they do indeed work, and they work very well. So in early 2013, just about 6 months after Jennifer and Emmanuel's original paper appeared, there were a series of papers that appeared demonstrating that these nucleases could be programmed and target and do genome editing in cultured human cells, including pluripotent stem cells, and I believe you'll be hearing from Chad Cowan later today. And our own contribution to this was we demonstrated that you could do this in single-cell embryos with zebrafish, inducing targeted changes in gene sequences of interest. So that's the introduction. Now what I want to do is give you an overview of what I'm going to talk about today. So we've been very focused on the issue of specificity for these CRISPR-Cas9 nucleases. And essentially, I'm going to divide the talk up into 3 parts. The first is I'm going to talk about how we define the specificity of these proteins. In the second part, I'm going to talk a little bit about our efforts to improve the specificity. And then in the last part, this is the unpublished part, I'll talk about our efforts to change or alter the specificity of the Cas9 nucleases as well. So let's start with define. So now, as I've just shown you, targeted genome editing is very, very simple with the CRISPRs. You just have to change the RNA. But almost immediately, we started wondering, what are the off-target effects, or what are the spectrum of off-target effects that one would see with these types of nucleases, where most of the recognition is being driven by this RNA-DNA complementarity. And of course, this is an incredibly important issue to address, not only for research applications to understand where these off-targets are occurring and to what degree and how frequently they're occurring, but in particular, if you're going to use these reagents for therapeutics, then one needs to have as complete an understanding of where these off-target effects may be occurring as possible. So, in our minds at least, the ideal method for finding off-target effects of nuclease would have at least 3 characteristics. So one is, obviously you want the coverage to be genome-wide, because you don't want to miss any potential off-target effects in the cells that you're modifying. The second requirement is that it really be unbiased. And again, this is a question of coverage to some degree. You don't want to make assumptions about where these off-targets may happen and therefore miss them. But you also want it to be unbiased in the sense that you don't necessarily want it to just run on a reference genome. Because especially as we think about doing this on individuals as a therapeutic going forward, not everybody obviously in this room has a reference genome. In fact, probably none of us do. And there are differences that can then lead to differences in the spectrum of off-targets. So you really want a method that is essentially customizable as well to the individual's genome. And then the last point is that we want it to be highly sensitive. And I think this goes to therapeutics in particular, where even a low-frequency off-target effect could have deleterious effects and actually could be sort of selected for in an in vivo context. In particular, when we worry about mutations that lead to transformation events and those cells being able to grow out and have a selective advantage. So the problem for the field has been that this type of method has really eluded the field. There hasn't really been a method that has all three of these characteristics. So what has been done? So one approach has been to take single-cell clones that have been modified and have the desired change of interest and simply do whole genome resequencing. And I'm going to ignore the technical issues associated with sort of the fact that you're culturing these cells and expanding them and the fact that you're probably acquiring mutations in the process of doing that. But just note that some people have done this in the literature and have found that they can't identify any mutations that are attributable to the expression of the nucleases. And on that basis, have therefore concluded that these nucleases don't induce off-target effects. But of course, you can't really conclude that from this type of experiment because you're not really comprehensively looking at what are all the different types of off-target effects that a nuclease can induce. In fact, all you've really done is show that for that particular cell, you don't see any evidence of off-target mutations. But that's very different than saying that the nuclease you use is incapable of introducing off-targets, and that in other cells that are modified by that nuclease, you might have an off-target. The other issue with this method is that, of course, intrinsically, by sequencing just a single genome, you're going to miss lower frequency off-target effects, right? Because the probability that you have an off-target effect in the same cell that you have an on-target is going to be somewhat proportional to the difference in frequency between those events. So if an off-target effect occurs less frequently, then you're going to have a lower probability of seeing it in a single-cell clone. If flipped around, let's say you have a mutation that occurs only once in every 100 cells, even if you sequence the genome of 100 cells, you still may not see that off-target alteration. So this is the problem, is that in order to be able to make conclusions about whether or not a nuclease is capable of introducing off-target effects, especially low-frequency ones, you would need to be able to sequence a lot of cells and a lot of genomes. Next slide. So the other approach that people have taken is really to do what we call focused next-generation sequencing approaches. So what they do is they say, okay, here's the target site. We know what that sequence is. We scan the genome using computational approaches. There's some programs that are out there that we'll talk about in a little bit that identify sites that are similar in sequence to the on-target site. And then people essentially amplify those regions out from cells, populations of cells that have been modified by the nuclease, and ask, do you see evidence of off-target mutations at those sites? And the answer is yes, you can find off-target sites this way. In fact, this was how we and others first demonstrated that you could get off-target effects in human cells using CRISPR-Cas9 nucleases. But of course, the problem with this is that it's a lot like the man who drops a quarter in the dark, and is illustrated in this cartoon. He's looking for it under where a lamppost is shining. A police officer comes along and asks him, did you drop the quarter here? And he says, no, I dropped it down 2 blocks down the street. And so the officer asks him, well, why are you looking for it here? And he says, well, because the light is brighter here. I think the light is better here. So if you think about it, that's really what this focused sequencing approach is doing. It's saying, here's the genome, and this is large dark space. And with our computational approaches and with deep sequencing, we're essentially shining the light on a handful of places and asking, do you see evidence of off-targets there? And the answer is yes, you can find them, as I just said. You can see these little red X's representing off-target effects, and you can see them under some of the places that you shine the light. But the question is, are there other places in the genome where you're not shining the light, where these off-target effects may also exist, and you're just not seeing them because you're not looking? Yeah. Characteristics I mentioned earlier, again, genome-wide, unbiased, and that is highly sensitive. We developed what we now call the GuideSeq method, which stands for genome-wide unbiased identification of double-strand breaks enabled by sequencing. And what this relies on is the observation that if you take a short double-stranded oligo that's been modified to stabilize it in cells, you can get very efficient uptake of this oligo anywhere where CRISPR-Cas9 is introducing a double-stranded break. So in the process of capture into that break, you essentially tag that sequence, That then gives you a handle from which you can do essentially one-sided amplification. So you can amplify only those genomic sequences in a unidirectional fashion that have this tag integrated in them. And I won't go through all the molecular biology here, but you can barcode the sequences, the fragments, so you can tell whether you're isolating the same sequence repeatedly as independent fragments or not. And then you can subject this whole thing to deep sequencing and map back to the genome and where these integration events have actually occurred. Okay? So, next slide. So, what this allows you then to diagram of the entire human genome. We're showing you here in green the location of the on-target site, and these red marks indicate all the places in the genome where we've captured these sequences because the tag has been incorporated. And so you can do two things. One is you can tally up the number of these red marks and plot them as histograms, and get some sense of how many off-target effects one is seeing. I'm going to come back to this data in just a second. So to test this out, what we did was do it with 10 different guide RNAs. And 4 of these guide RNAs we had previously identified by that shining the light on certain parts of the genome approach, some off-target sites. So it provided us a nice internal control to get a sense of whether we were able to at least identify all of those sites. Another set of these sites, the ones on the bottom, were 4 sites that had previously been subjected to a genome-wide ChIP-seq protocol using a catalytically dead version of Cas9 known as dCas9. And from that, off-target binding sites of the guide RNA associated with that Cas9 had been inferred. So again, we did this because we wanted to be able to compare the results of guide-seq with what was seen with this genome-wide ChIP-seq. And then the other thing just to notice is that we did this in 2 different transformed cancer cell lines. Okay, so as I said before, you get these genome-wide profiles, and this is just a histogram again showing the number of off-target sites that were identified for each of the 10 different guide RNAs we tested. I'm showing you here in red all the sites that were previously found for these 4 different guide RNAs, and I'll just tell you that we found all of the off-target sites that had been previously identified for those 4 guide RNAs, suggesting again the comprehensiveness of this approach. But of course, it's interesting to note that in addition to those that were previously known, we found a whole bunch of additional sites as well. The other thing to notice is there's a tremendous amount of variability in the total number of sites, off-target sites that one finds going from guide RNA to guide RNA. Some of them have very few, some of them have a lot. Indeed, this one here, RNF2, doesn't appear to have any off-target sites, at least judged by the level of sensitivity that we had with the guide-seq approach as we practiced it here. We did look to see whether there was any preference for these sites to be located in a specific type or regions of the genome, so in introgenic, exonic, or intronic regions. And the upshot of this analysis is that there is no preference for one type of the genome over another. So they appear to be roughly equally distributed across different areas of the genome, even accounting for the fact that these different types of regions are not equally represented in the genome as well. Next slide. So the other thing that you can do, in addition to sort of just tallying up the number of off-target sites that you find, is you can also get some sense of the frequency at which those sites are being broken. And so the reason you can do this is you're isolating typically for any given site multiple sequencing reads. And as I said to you before, we can tell whether these are independent isolates of different fragments or not because of the barcoding scheme that we use in doing molecular biology. And so when you map these reads back to the genome, you'll see things like this, where you have multiple reads mapping around a particular break site. And you see them going in both directions, both because the tag can integrate in both directions, but also because we prime off of that tag in both directions as well. And so that's why you see 4 types of colors in terms of the type of, in terms of the type of integration that you see. And the nice thing is that by looking at the junction between the genomic site and the tag, you can map essentially to the nucleotide level where that break is occurring. So you can do these plots, histogram plots, that show you at a given location what are the number of sequences you isolate that have a junction at that point. And this essentially allows you to identify where the break is occurring. So you can see here, this is a sequence that's a PAM sequence, and 3 bases in, you get this cut site that's the predominant cut site for that particular target. So a prediction from this is that by counting the number of reads you get for any given locus, that in turn should reflect the frequency with which these things are being broken in the cell, right? And so we wanted to test that formally. So what we did was we took all the sites that we identified from guide-seq for 5 different guide RNAs. And then we just reran the experiment. So we just expressed the nucleases and the guide RNAs, but without now introducing the tag into the cells. And then we used focused amplification and sequencing of those targets and asked what was the indel frequency that you see at those sites. And so with that data, then you can do scatter plots for each of the sites. And what you can see here is this is the number of guide-seq read counts we got, so the number of times you isolated a particular sequence from guide-seq, versus the indel frequency here that you saw at the same site. And you can see there's a nice correlation between the number of read counts and the frequency of mutagenesis at those sites. So the upshot of that is that the guide-seq read counts are a surrogate for the frequency with which the sites are being broken. The other interesting thing to notice here, though, is that you start bumping up against this indel frequency of 0.1%, which is really about the error rate of the next-gen sequencing platform that we used for these experiments. And so below that level, it becomes hard to tell the difference between whether an indel is being introduced by the nuclease or whether it's just error in the sequencing itself. But we do see a lot of things that sort of bunch up down at essentially that point, which we just labeled as 0 here. And you'll notice that most of them have low-level guide-seq read counts that fall below 100. So we think that these things that are bunched up here probably represent bona fide off-targets because we're capturing sequences at them. But we can't tell whether we're actually introducing mutations when we just do the nuclease-only experiment because, again, we bump up against this error rate of the sequencing. So the upshot of that is that we think guide-seq is able to detect sites that are broken that would yield an indel frequency of 0.1%, but probably even lower than that, and it's just hard to tell by sequencing that these are actual sites. Remember that with guide-seq, we don't fall into that problem of the error rate because we're not looking for an indel. We're actually looking for the captured sequence. And so we don't have that same problem that you do when you're just looking for indel mutations. Okay. This is a slide that you can't possibly read, but it's just to show you for the 9 different guide RNAs for which we isolated off targets, what those sites look like. So at the top is the intended sequence. And underneath, I've listed all of the off-target sites showing you in colored squares where mismatches occur. The PAM in every case is located here on the right-hand side, and so you always expect this one base to be variable because that's the end in the sequence. So the things I want you to notice from this are that you see a lot of mutations. They're scattered throughout the spacer sequence, right? So they're not necessarily limited to one side or the other. So people talk a lot about the seed sequence on the 3' end, but it's clear from this type of analysis that the seed sequence is not immune to having mismatches in it. It's hard to find them here. I don't know exactly where they are, but there are sites that have up to 6 mismatches in their protospacer sequence. And we also found one site where there's a so-called bulge mutation, where there's a mismatch between the guide RNA and the DNA that results in one base being bulged outward. The other thing to notice is that we do see some alternative PAM sequences. So people had previously identified NGA and NAG PAMs, and we found those, but we also found NGC and CG, and I believe in one case an NGT as well. I can't find it for you right this second. So very complicated is what this means. It's hard to predict where these off-targets are happening. So I told you before, there are these computational programs that will take your on-target site and try to predict for you where the off-target sites occur. So we were very curious to compare the results from guide-seq with the output from these programs. One is the so-called CRISPR design tool, and these are Venn diagrams showing you in blue what the sites that we got from guide-seq, in yellow the ones predicted by the program. First thing to notice is that the program does not capture all of the actual bona fide off-target sites, and it has this bit of this sort of false positive problem, if you will, that it seems to overcall a lot of sites that may not actually be bona fide off-targets. The Boutros group in Germany have a similar tool that they call eCRISP, and you can see here that you get a similar sort of result. Again, misses a number of sites, tends to overcall sites that are not bona fide off-targets. Remember earlier I told you that 4 of the guide RNAs we used, people had done genome-wide ChIP-seq with before. So these are just Venn diagrams, again, comparing guide-seq with what the ChIP-seq show. And here it's very clear that there's essentially no overlap between the 2 types of sites. I've told by our biostatistician collaborator that this little overlap here is probably just due to chance, essentially. And you can see from these plots that plot the number of mismatches you see in the off-targets of guide-seq versus the chip-seq sites, that they are very, very different. So the guide-seq sites have far fewer mismatches than the chip-seq sites do. So these sites, I think, generally now it's accepted in the field, they're probably not bona fide off-target sites of the nuclease. Whether they represent anything biologically relevant for the dead Cas9 in terms of this finding, I think remains an open question. Okay, so believe it or not, even though I've just spent like a half an hour on this, there's actually a lot more data in this paper, that a lot of interesting findings, where we're capturing nuclease-independent breaks in the cell using guide-seq, and we also use the information that we get from guide-seq to show the translocations occurring between different on-target and off-target sites, but off-target and off-target, as well as between the nuclease-induced sites and the non-nuclease-induced sites as well. So I refer you to the paper for additional details. Okay, so now I want to talk briefly about improving the specificity of nucleases. So if you think about the original streptokinase Cas9 as sort of a first-generation CRISPR-Cas nuclease, people in the field have developed other platforms to try to improve the specificity, knowing early on that indeed you could get high levels of off-target effects for some guide RNAs. There are three approaches that have essentially been described in the literature. I want to focus initially on this one, which we call the truncated guide RNA approach, in which we described last year in the paper. So the idea here is that you can increase the specificity of Cas9 by shortening the region of complementarity at the 5' end of the guide RNA. So I told you before that people will typically use 20 nucleotides. We found that by shortening it to 17 or 18 nucleotides, you can actually increase the specificity of the guide RNA Cas9 complex. Now you may say that's a little counterintuitive. If you try to improve specificity, why would you actually shorten the guide RNA? And so the reason that we did this was some experiments that we did early on, where here's a regular standard full-length guide RNA with 20 nucleotides of complementarity, and it's very happy to cleave its on-target site. But what we found was that we could march in and deliberately mismatch bases at the 5' end of this guide, going up to 3 nucleotides, and still see full activity of this complex on the on-target site. But interestingly, in every case we looked at, once you mismatch the 4th base, then this on-target activity essentially plummeted to 0. So what that said to us was two things. One is that the original complex probably has more binding energy in it than it needs in order to be able to recognize the original on-target site. The other thing it says is that if you shorten this guide RNA by up to 3 nucleotides, it's now probably energetically poised to have just enough interaction energy to cleave the on-target site still. Because, and I say it's poised because we know that if you shorten it by 1 more base, you essentially kill the activity of the guide. So because it's poised, what we predicted was this should then make this guide RNA Cas9 complex more sensitive to mismatches at the interface. So that if it encounters an off-target site, then it's very, very likely that it wouldn't be as active at that site because, again, it was energetically poised, and now with the mismatch, it's not going to have enough energy to do what it needs to do. Does that make sense? Okay. So we tested whether this was true. So I showed you before, we had on-target sites, so here are 3 of them, and we had some known off-target sites for each of them. And what we did was use deep sequencing and PCR to compare the rates of indels that you get with the standard length guide RNA for each of these sites and a truncated guide RNA. So with the standard length guide RNA, you see high levels of on-target mutagenesis and high levels of off-target mutagenesis as well, which was not unexpected. But look what happens when you use the truncated guide. Now, in every single case, you reduce the frequency of mutagenesis. It doesn't go to 0. There's still a few sites where you see some residual mutagenesis, but in every case it goes down. But in every case, the on-target remains essentially unchanged. Okay? So validating this hypothesis that by shortening the RNA, you can retain full activity at the on-target site, but reduce the off-target the frequency of mutagenesis at off-target sites. Now, when we published this paper about a year ago, some of the reviewers gave us a hard time because they said, well, how do you, this is known off-targets using the looking under the lamppost approach of these guide RNAs. And yes, you've reduced those, but how do you know that in the process of truncating the guide RNA, you haven't increased or created new off-target effects that you're not looking at here, and that weren't off-targets for the original? And so, of course, we could not address that question originally, but now with guide-seq, we can. And so this is just, again, histogram plots showing you total number of off-target sites
And this is the same results I showed you before, 3 of those guide RNAs with known off-targets. You can see you get very high numbers of off-targets. When you do the same experiments, same 3 sites, but now with the truncated guide RNA, look what happens. Again, you drop the total number of off-target sites for each of the guides. So this suggests that then what we've done is not only decrease the frequency of off-target sites that were attributable originally to the original full-length guide RNA, but we really haven't created a large number of new off-target sites in the process. So we were able now to show that indeed on a genome-wide scale, the truncated guides are more specific. This is just a little easier to see now because there are fewer sites. Same kind of plot I showed you before. It shows you that the off-targets that we do find have fewer numbers of mismatches in general than what we were seeing with the full-length guide RNAs. Again, consistent with this idea that we've created guides that are less tolerant to mismatches, and therefore you're not going to see sites that have large numbers of mismatches. There are these other approaches, so paired Cas9 mutases, which many of you have heard about, I'm sure, originally developed by George Church and then further elaborated by Feng Zhang. There's a similar approach that Li and David Liu also described, but using true dimeric nucleases where we use dead Cas9 fused to dimerization-dependent FOLC1 nuclease. I'm not going to go into detail about either one of these methods. I just refer you to this paper for the dimeric FOLC1 dead Cas9 fusions. I will say that we've tried to do guide-seq with paired nucleases, and we can capture the tags into the double mixed sites. We do see capture into the sites cleaved by our dimeric FOLC1 Cas9 fusions, but we're still trying to optimize the efficiency of it because the capture isn't quite as efficient as it is with the wild-type Cas9 enzyme. So for the time being, the only improved specificity platform that's really been validated on a genome-wide scale is the truncated guides, but we're trying to work out whether we can use guide-seq to assess these other platforms as well. All right, so I'm trying to do pretty well. So take-home messages so far from the talk. One is that guide-seq is a genome-wide unbiased and a very sensitive method for being able to detect off-target sites. We believe that to date it probably is the best method and the most sensitive method. There have been a few others described in the literature, but we believe that all of them will suffer from sensitivity problems because the way they tag the sites is a lot less efficient than the way we do in our process. It's not biased. We don't make assumptions about where the sites are. We're just incorporating tags into wherever breaks are. So it's not necessarily confounded by having to have a reference genome sequence, meaning that it can be done in a personalized fashion. And then we have the sensitivity is very high. We think we can detect sites that are mutagenized at frequencies of 0.1% or even lower. The other things to note are that we think that high specificity does appear to be possible. So there are some guides that do appear to have very few off-targets. Remember, there was even one where we saw no off-targets. We also were able to use guide-seq to show that the truncated guides have improved specificity on a genome-wide scale, and also our results demonstrate the limitations of the computational approaches that are out there for identifying off-targets. And really, I think the take-home message at the end of the day is that if you care about off-targets and you care about identifying them, then you're going to have to use an experimental approach, at least for the time being, to identify them. Okay. So now I want to talk about the last bit, which is the unpublished work, and that's shifting gears a little bit. So now I'm going to talk about changing the PAM recognition specificity of the Cas9 protein. So this is a schematic of a gene that one of our collaborators wanted to target. They wanted to do HDR, at this particular nucleotide here, making a CBT type change within the coding sequence. So of course the way that you would do that is design a donor molecule that has the change that you want, as we described earlier, and then really what you want to do is put a break either right at the site of the change or certainly within about 10 to 20 nucleotides of where that change is. Otherwise the efficiency of the process, which isn't great to begin with, becomes very, very low. So the problem that we encountered with this collaborator's project was that when we looked for 20 nucleotide full-length guide RNAs with strep pyogenes Cas9, the closest thing we could find made a break that was 53 nucleotides away. Now you can increase that targeting range a bit by using truncated guide RNAs, so because you can do variable different lengths, it gives you more possibilities, but even doing 17, 18, and 19, still the closest thing we could find was 41 nucleotides away. And this is a reflection of the fact that the genomic sequence is obviously not completely random. You're looking for that NGG sequence, and sometimes you're just in a region of the genome where that is just relatively poorly represented. And I should say that we have about a dozen of these different examples where the collaborators, we were unable to target closely enough to where they wanted to be in order to get the change. And so again, I've just shown you this example for HDR, but it also is the case even when you're trying to do NHEJ, particularly for small genetic elements. So we have collaborators who want to knock out microRNAs, want to knock out splice sites, want to knock out transcription factor binding sites, and even some people who want to do knockouts of these short ORPs, or I think they're called nanopeptides as well. And then the other thing that sort of made us think about trying to change the specificity of the PAM was that PAM recognition is believed to be very, very specific. So one way to achieve allele-specific editing might be to place the difference in sequence between two alleles within the PAM recognition sequence. So if you can do that, then you might be able to discriminate between one allele versus the other based on the fact that the PAM discrimination at Cas9 is usually very, very good. The problem is, of course, that essentially your PAM is NGG. There's only a very limited number of cases in which you're going to be able to do that. So having Cas9s with more Cas9s with different PAM specificities would allow you to better be able to practice this type of allele-specific alteration method. So that's the rationale. So fortunately, there was this structure that was solved by Martin Yinnick and his colleagues of Cas9, strep Cas9, a guide RNA, bound to its target site. And what this revealed is that recognition of that PAM sequence, so the two Gs in the PAM sequence, is mediated by two arginine side chains of position 1333 and 1335 in the Cas9 protein. So initially, we naively thought, well, okay, we know this arginine contacts this G, we know this arginine contacts this G, we know from other protein DNA structures that recognition of an adenine can be mediated by glutamine with very high efficiency. So maybe if we substitute the arginine here with the glutamine, or the arginine here with the glutamine, we can change the specificity so that you're now recognizing an NAG or an NGA PAM. So this is an experiment with wild-type Cas9 that just shows you that you get very good cutting when the PAM is NGG. You get a little bit of cutting when it's either NAG or NGA, which was already known, and I showed you that from the guide-seq results as well. And you really don't get any cutting when it's NAA PAM. So we thought, okay, let's make these mutants. Let's change 1333 to Q, 1335 to Q, and let's make a double Q mutation. And maybe we'll be able to recognize NAG, NGA, or NAA. And of course, as you can see from this experiment, the answer is that we don't recognize anything anymore. We just kill the activity of the protein. So it doesn't recognize NVG, but it also doesn't recognize the new site of interest. So what is said to us is, okay, maybe there are some other changes that have to occur in the protein in order to be able to identify a mutant that recognizes the new sites. And just looking at the crystal structure, you can see that recognition is complex. There are other residues that appear to, particularly in this region here, the lock region, may play an important role in mediating recognition. So it wasn't an unreasonable hypothesis, we thought, that perhaps you needed other changes in the protein. But designing this is not trivial, as so we needed to have a way of finding these mutants. So what we did was we randomly mutagenized the PAM interacting domain part of Cas9. And we used a bacterial selection system that was based on one developed for meganucleases many years ago by Hyunmin Zhao's lab and David Liu's labs. And so this involves a plasmid that encodes a toxic gene, and you put a target site of interest for a nuclease on the same plasmid. So if you introduce into the cell this plasmid and a plasmid encoding a nuclease capable of cleaving this site, this will become linearized, linear DNA is degraded in E. coli, the toxic gene will no longer be expressed, and a colony will survive on a bacterial plate. So what we did is we put a target site here that had an NGA PAM. We decided to go after that one first. And then we took Cas9 that had a Q at the 1335 position, and then we randomly mutagenized that, introduced this library in, and asked, can we get surviving colonies? And the answer is you can, and when you sequence those, you get this. So here is the original mutation that we introduced, and then you can see that there are certain positions throughout at which you get different alterations. So there appeared to be a couple key other residues where we were repeatedly getting mutations. And so you can then go back and do some additional combinations of different mutations and testing, and I'm not going to go through the whole detail. It was an iterative process that we had to go through a few times. But the upshot is we ended up with, in the end, something that we call the NGA variant. So this is a strep pyogenes Cas9 that now appears to recognize an NGA PAM rather than an NGG PAM. So this is mutation data on endogenous human gene target sites using different guide RNAs and this NGA variant PAM. And you can see that it's very robust. You get very, very high frequencies of mutagenesis. I will say that there does appear to be some preference for a G at the base right after the NGA. This is the most highly cleaved type of site. There's good cleavage still of NGA-T and NGA-A, and somewhat weaker cleavage of NGA-C. Going through that whole process with a different rational substitution and a different mutagenesis round and so forth, and a different sort of combination of mutations, we also generated what we call the NGCG variant. So now, this is a strep pyogenes Cas9 that instead of recognizing NGG, recognizes NGCG as the PAM. And again, this is human cell endogenous gene data demonstrating that this is robust and you do get very nice mutagenesis and cleavage of the site. Okay, so we wanted to ask, for these sites that we were able to robustly target with these different SBCas9 PAM variants, are you still able to cut these sites using wild-type SBCas9, right? Because if you could do it with wild-type SBCas9 still, then obviously these are less useful. But as you can see from these graphs, the answer is SBCas9, wild-type SBCas9 really has no effect, or very little effect, on any of these alternative PAM sequences. So this is a demonstration that you really need to have these variants in order to be able to efficiently target these new types of PAM sites. Okay, so going back to that example I showed you earlier of our collaborator, now we were able to, with the new NGA PAM, that wasn't just me, right? I was up late last night working on things, so I just want to make sure it wasn't me. So now we can target right on top of the change that we want and introduce actually a break very, very close to where the change is. So this is just a demonstration that with the new variants, we're able to get to sites that we previously couldn't get to. So all of this, though, is just to say that it can cleave at the on-target site, right? But then an important question is, what is the specificity of these new variants, right? So maybe we've just created something that is able to recognize the new sites, but maybe also recognizes all kinds of other PAMs in the cell, and therefore has worse specificity. So fortunately, again, we have guide-seq. So we did guide-seq on a variety of different guide RNAs, both with the NGA PAM and with the NGCG PAM. And so these are histogram plots of what we found. So these are the 8 guides that we did with the NGA variant. These are the 5 guides that we did with the NGCG variant. And then this is just showing you the same data I showed you before, the 10 guides with the wild-type strep pyogenes Cas9. So what you can see is the NGCG variant is highly specific. There are very few off-targets that we find. The NGA variant certainly looks no worse than what you see with wild-type Cas9. So what we conclude from this is that we're probably not doing any worse than we were with the original SBCas9. And indeed, in particular for the NGCG variant, we might even be doing a little bit better. I'm a little hesitant to make that conclusion definitively because I think we need to look at more guide RNAs with these different variants. But certainly this first pass suggests that these things are at least no worse than the original protein. And this is just showing you what the actual sites look like. Same type of plot I showed you earlier. So all of these here are for the NGA, all of these here are for the NGA variant, and these are for the NGCG variant. And you can see that as you saw with the wild-type pyogenes Cas9, you do see mutations sort of distributed throughout the spacer sequence. And actually, if you look at the PAM sequence, we did a pretty good job of recovering the PAM that we intended to get. So I'm showing you a 4th base here at the end, that's why there's some variability. But you can see the preferences that I mentioned earlier at the 4th position. And with the NGCG variant, it is quite actually specific here for the target site. And it's not surprising, right? Because the PAM is actually longer now. So the site is therefore, the target site is actually longer. So you would expect that to be a little more specific in the context of the cell. All right, so in conclusion for this part, with these engineered variants now, to put some numbers on it, we've essentially doubled the targeting range of wild-type SBCas9. So this is a pie chart that's showing you the sites that you can find in the genome that have an NGG PAM. And if you look at all the NGA PAMs and this little sliver that's the NGCG PAMs, which is not only because it's 4 bases long, but apparently because this type of triplet is underrepresented in the human genome. you get this number of total sites. And as you can see from the pie chart, we essentially have more than double the targeting range. These variants show genome-wide specificities that are at least comparable to wild-type Cas9. And perhaps more importantly, this is a really, I think, a really important proof of concept that one can actually modify the current specificity of Cas9. And perhaps even more broadly, using the same types of selection systems I've described to you, it suggests that one can also modify other functionalities of Cas9 as well. Should be relatively simple to create now nicases and dead Cas9 variants because we built all this stuff in the context of asparagines Cas9, which is, again, the most widely used Cas9. And we think that some of these variants now provide a starting point for being able to go through that whole engineering process again, but now trying to engineer Cas9s that recognize even more divergent PAMs. So remember the MAA PAM that I told you in the beginning, we wanted to get, but we weren't able to get. And so we think that, again, these things that we've got so far give a nice starting point. Now, I know many of you will want to look at your genes of interest to see whether or not you can target with our new variants. We've developed a new software program that we call Cas-Blaster. So it's at www.casblaster.org. The site was launched about 2 weeks ago. And it allows you to do not only Pyogenes wild type, but also our variants, and also Streptococcus and Staph aureus, which other people have shown work well in human cells. It also allows you to do those dynamic RNA-guided Cas9s if you want to do it as well. We have deposited, or are in the process of depositing the plasmids for these variants with AdGene and hope that they'll be available around the time of the paper actually appears. So I'll stop there. I need to acknowledge the people who did the work. The PAM variant work in particular was done by Penny Kleinsteiger, Michelle Prue, with help from Shendar Sai, from Wei Chokar, and Liu Nguyen, also from our collaborator Martin Arden. The guide seed work was done by Shendar Sai and New as well, again in collaboration with Martin Arden and our collaborator John Iafredi for some molecular biology. And then in terms of the truncated guides, that was work done by Yantan Tu and Jeff Sander, and then the guide seed for the truncated guides was done by Shendar Sai. So again, here's the address for the software if you're interested in trying to find whether the variants are targetable for your sequences of interest, and we hope to, as I say, make the plasmids available through AdGene. And I need to thank the funders at the bottom for their support as well. So I'll stop there. Happy to take any questions.
If you have a question for Keith, we would ask you to use a microphone. We are recording this, so we do want to make sure that everyone can hear those questions. And Keith, I want to thank you. That was fantastic. And I think the other thing that I want to thank you for is this is what's really nice about this field is the sharing of information and reagents. And I think that will be instrumental in pushing it forward really fast. So thanks for that. Questions for Keith?
Hi, Keith. How are you? Just curious, because even the modification you made, for the new PAM sequencing, we're right for distribution bar. So I'm just trying to get a sense of your feeling whether we should move into the truncated bar,
Yeah. So we don't have a ton of experience with the truncated guides with wild-type Cas9, but we've heard from people who've done larger scale work with it that it is very robust in terms of on-target activity. I don't think so many people have looked at the off-target by IC, but in terms of getting good on-target activity, it seems to be generally pretty consistently high. We have done some work with the truncated guides with the variants as well. And it also looks very good. We just have added publication quality for this one. But we do offer on the website the possibility for the truncated guides as well.
But do you have any call on you?
I personally would use the truncated guides because I think you're going to have fewer off targets with them. I think it's very clear that you're going to have fewer off targets, and there's no real downside to using them because in general the efficiency is quite high. Yes.
Thank you very much, Dr. Ernesto. I just wanted to ask you, have you tried using the eggs for the guide seq? to be, so in other words, are you sure you're not being biased, you're just using one particular type?
Yeah, no, it's a good question. So the answer is we have not described any for using different tags. So the tag we use is 34 base pairs in length. It's designed to be as orthologous as we could make it relative to the rest of the genome sequence. We've done some subsequent optimization to suggest that that length is actually pretty good for capture. You can't get any shorter than that, certainly if you want to prime in both directions. It's hard to make something shorter than that. And in terms of bias, we don't see any evidence of bias, and we don't believe there's any bias in terms of where it's being captured. We'd look to see if there's any evidence of microhomology in the sites that the tags being captured into, and we don't see any evidence of that. We can't completely rule out that there's some degree of bias, so I agree that doing a different tag would get at that issue. His for slides have just been, we barely kind of got this stuff together and published in the time frame we did, so we're now going back and trying to do more of these studies that optimize the tag. And ultimately, I think some kind of in vitro equivalent to guide-seq will help to address some of these questions.
What is the off-target compared with in cancers versus fragment cells?
Yeah, that's also a good question, and also something that we're looking at. So you might expect that the rates of capture and the rates of genesis, even at the end-up level, might be higher in transformed cells, which depends on sort of what their genetic background is. But I don't know the answer to that. That's something that we need to look at as well. Again, it's very early days for us to see this. But we're now in a position to be able to do that experiment, those types of experiments. The other experiment we can do that we haven't done, but I think will be very interesting, is to follow the kinetics with which these off-targets arise in a population of cells using anti-seizure cells.
Did you see the future as being a collection of different variant Cas9, each specific for a different PAM of 4 nucleotides, so that we would have a choice of which one we would use?
Yeah, I do actually. I think it was a tremendous amount of work just to make these 2 variants, but part of it was we were feeling our way through it. I think now going back and recycling it, and then also using some of these as starter points for engineering an even greater orthogonality, I think will make the process faster and easier than it's been. But yeah, I envision going forward that there shouldn't be any targeting range limitations in the future. The other thing you can imagine is, other people are starting to characterize Cas9s from other orthologs from other species. So, for example, Fung's group has recently published on Staph aureus Cas9, which has an advantage much smaller, and appears to be very, very robust. In our hands, certainly human cells is very robust as well. So you could imagine engineering on those different platforms as well, using a very similar approach to what we used here. But structural information would probably be necessary just because the structure-guided mutations were important for getting us to where we were with certain pathogens.
So Keith, I'm going to take the liberty to ask the last question. When Young and I were postdocs, we created a targeted translocation with 3 locks. Do you think there might be a future to, you mentioned that we see translocations with the off-target, to actually do targeted translocations with Mr. Cas9?
Oh, absolutely. I mean, those have been published already, too. So there are groups that have done it already. I think the frequency is still relatively low. Even in our hands, looking for these off-target, on-target type translocations, the frequencies are relatively low. But they definitely can be induced, and I think people are probably looking at ways to increase the frequency of those events.
Thank you for the nice talk.